How are viruses used as delivery vehicles in gene therapy? Imagine a tiny messenger that can sneak into cells and deliver important instructions. That's what viruses do naturally, and scientists have learned how to use them for healing. Viruses are great at entering cells and delivering genetic material, which makes them perfect tools for gene therapy. But instead of causing disease, scientists modify these viruses to carry helpful genes that can fix or replace faulty ones in a patient's body. To do this, they remove the parts of the virus that cause illness and replace them with therapeutic genes. These modified viruses are called viral vectors. When introduced into the body, they act like delivery trucks, transporting the new genetic instructions directly into specific cells. The virus's outer shell, called the capsid, protects the therapeutic gene and helps it get inside the target cells. Different types of viral vectors have unique features that influence how they interact with the immune system and which cells they target. There are two main types of viral vectors used in gene therapy. The first is called integrating viral vectors. These include retroviruses and lentiviruses. They insert the therapeutic gene into the cell's DNA, which means the gene can work for a long time. This is useful for diseases that need ongoing treatment. But there is a small risk because inserting new DNA can sometimes disrupt important genes in the cell, which might cause problems. The second type is called non-integrating viral vectors. Examples are adenoviruses and adeno-associated viruses. These deliver DNA that stays separate from the cell's DNA, so the effect is temporary. Adeno-associated viruses are especially popular because they cause little immune response and are less likely to cause illness. They are often used in treatments for diseases like inherited blindness or certain muscle disorders. Scientists also modify the surface of these viruses to target specific cell types. By changing the proteins on the virus's surface, they can make the virus more likely to infect only certain tissues, such as liver, muscle, or brain cells. This targeting helps reduce side effects and makes the therapy more effective. The immune system plays a dual role in viral gene therapy. It can recognize and attack the viral particles or infected cells, which might reduce the therapy's success. To prevent this, scientists design vectors to be less visible to the immune system. They remove viral genes and choose vectors that don't trigger strong immune responses. This careful design helps ensure the therapy works well and lasts longer. Delivery methods vary depending on the disease and the target tissue. For some conditions, doctors inject the viral vectors directly into the affected tissue to limit immune activation. In other cases, they modify cells outside the body and then reintroduce them, known as ex vivo therapy. This approach allows better control over immune reactions and makes sure the therapeutic genes are delivered accurately. In summary, viruses are natural delivery agents that scientists have adapted for gene therapy. By engineering these viruses to be safe and specific, they can deliver therapeutic genes effectively. Managing immune responses is also key to making sure these treatments provide lasting benefits. This innovative use of viruses opens new doors for treating many genetic and acquired diseases.